All right, good afternoon, everyone. I'm going to go ahead and get started on time here. Uh, Welcome to the first grand rounds of the new year. Today, I'm going to talk about the anatomy and embryology of the cardiac conduction system. And remember to enter the CME code 45496 to show your attendance. So if the keyboard's going to work for me. I guess we're not getting started on time. There we go. Okay, so today I'm going to talk about kind of the pertinent features of the gross anatomy of the heart in relation to the cardiac conduction system. Spend a couple of slides defining the cardiac conduction system, and then a lot of the time on the embryology of the cardiac conduction system, as well as some of the histology and function of the components of the cardiac conduction system. The objectives of the talk kind of mirror the outline here. One, understand the gross and microanatomy of the cardiac conduction system. Two, recognize the key role of asymmetry in the development of the heart, the conduction system, and particular today, the sinus node. Three, recognize the cardiac conduction system is not neuronal in origin. And four, understand that the working myocardium and the cardiac conduction system diverge early in development. So a little brief timeline of the cardiac conduction system and when things were discovered to kind of give you some background. The first ththing actually discovered was the Purkinje fibers in 1839. Followed by the identification of the sinus venosus as the pacemaker in the embryo by Gaskill. And then the first EKG recording was actually by Waller with the Lippmann electrometer. The AV bundle was discovered by Hiss in 1893. Wenckebach actually first described his group beating pattern named after him by looking at arterial and jugular pulsatility. The well-recognized string galvanometer by Einthoven as the first EKG in 1901. 1906 was a particularly very busy year for development or discovery of things in the cardiac conduction system, and started with the sinus node discovery by Keith and Flack, the AV node by Tawara, and identification of Wenckebach's group beating on the string galvanometer, and also the identification of what we now know as Mobitz 2 by Hay, all in 1906 there. And then Lewis confirmed that the sinus node is the pacemaker of the adult heart in 1908. And lastly, a paper by Mobitz summarized Wenckebach and Hay's work, and that's why we call it Mobitz 1 and Mobitz 2. So I find that you can never review anatomy enough. So most of my slides here kind of come from Heart and Coronary Arteries by McAlpine. And I'd highly encourage everyone to look at this atlas if you've never done so. It has excellent pictures and text that goes along with it. And kind of supplementing some of the conduction stuff, I've included some figures from Ho and Ernst. In the opening page of the right atrial chapter, McAlpine says a successful defense for the use of eponyms in anatomic description is impossible. And so I will try to use generic names where practical here. So the first slide here is of the external view of the right atrium. In the middle, we can see the pectinate muscle bounded by the terminal bundle and the anterior muscle bundle. And adjacent to that, we see this smooth supravalvular lamina, which will come up in future slides here. In the posterior of the right atrium, we see this smooth sinus venarium that comes from the sinus venosus. Taking this view and flipping it around from the inside, we see the vena cava on the screen right. That terminal bundle along the right side, which is sometimes called the crista terminalis. We see the nice pectinate muscles on the lateral free wall. And then in the IVC, we see this little thin membranous non-muscular valve of the IVC, also called the eustachian valve. And that's supravalvular lamina is adjacent to that tricuspid valve here. And two things to note is that the vena cava don't point directly at each other. They actually kind of direct inward to point the blood flow at the tricuspid valve. And this posterior atrioventricular sulcus is where we ablate for typical flutter. So I'll show that coming up. Taking a real oblique cut through here through the intraventricular septum, the tricuspid annulus, and the lateral pectinate muscle on the side, we really expose that terminal bundle, the crista terminalis. And we see this encircled the fossa ovalis where we use transeptal puncture. And then the valve of the IVC continues along into the atrial septum as the eustachian ridge. And this picture doesn't show it very well, but the os of the coronary sinus down here with the valve of the coronary sinus in front of it. Taking a short axis cut through the coronary sinus, we again see that valve kind of redirecting blood flow towards the ventricle. and really kind of poses a little bit of a barrier in accessing the coronary sinus with catheters. And as McAlpine labels it, the mitral valve, the superior and inferior commissures, that the inferior commissure here is really kind of adjacent to the AV node. The AV node's not in this plane, but I just kind of marked it here for your reference, and it's adjacent in with this non-coronary cusp. And in future slides, I'm going to talk about the right fibrous trigone, and that's kind of not well shown here, but it's the triangle of tissue between the right coronary cusp and the non-coronary cusp, which is continuous with the membranous septum, which is really important in understanding the anatomy of the conduction system. Taking the kind of four-chamber view in the inferior segment here and zooming into this white box, we really get a close-up picture of the lateral RV wall and really seeing this right atrioventricular groove. And we see this smooth supravalvular lamina comes up and forms the atrial wall of the groove. And the right ventricle wall comes in, and the annulus of the tricuspid valve is really inset from the surface of the heart. And of course, in that groove is the right coronary artery and small cardiac vein. So if you're standing at the apex and looking towards the base of the heart, this would be cut at kind of a 9 o'clock position. If we go to a 6 o'clock position, like in this next photo, you really see the annulus of the tricuspid is just kind of suspended here in very, very thin tissue. And we see the supravalvular lamina is just a really thin layer of tissue here. And in the background, we see the valves of the IVC and the coronary sinus. And so when we ablate for typical flutter, we really have to bring that catheter in and kind of hook it around to really kind of get back in this little pocket of this sulcus back here. Taking a superior view of the atria here, we see that the ventricles go off screen to the bottom. The aorta is reflected, revealing the superior wall of the left atrium. And behind that, spanning the atria, is the interatrial muscle bundle. And in this bundle, 3 and 4 have an electrical connection, sometimes called the Bachmann's bundle. And 1 and 3 have a sinus node that originates off the circ that either goes posterior or anterior to the appendage and traverses through this bundle over to the sinus node. The crux of the heart, crux Latin for cross. This kind of implies the picture on the left here that the chambers meet up to make a kind of cross or plus shape, but in actuality, it's nothing like that. And we see that the RV ostium is apically displaced from the LV ostium, and that's where we get the 1 centimeter when we're looking for Epstein's anomaly. And the interventricular septum is not aligned with the interatrial septum, so it makes this kind of dog leg shape. And this opens up for the possibility of communication between the LV and the RA, which is kind of the 5th type of VSD called the Gervais defect. And as I show later, the AV bundle will come up, cross over the crest of the muscular septum, and cross into the left ventricle through this area. And sometimes this potential space where these 4 chambers meet is called the inferior pyramidal space. So looking at an actual specimen here, we see that the middle cardiac vein has been ligated. There's a needle poking into this inferior pyramidal space and probably poking into the AV node itself and reflecting back that right atrium. We see a right PDA, a left PL system, and a left AV node artery. And here we can really see how that crux doesn't really hold up in shape. So we see the LV ostium, the interatrial septum, the RV ostium, and the interventricular septum in this diagonal connection, which looks nothing like a cross to me. So taking this view and kind of rotating around and getting rid of the right atrium, we see something interesting here that I didn't quite appreciate, that the right atrium actually wraps around the left atrium similarly to how the RV wraps around the LV, and kind of outlining that with a purple line here for you. The next picture will be, again, rotated a little bit further. Again, the RA attachment to the LA is kind of wrap around. And here is one of the first 2 pictures really showing this membranous septum that I really think is important for understanding everything here. The aorta is in the background here showing the non-coronary or right coronary cusp. That right fibrous trigone, which I'll show in the next slide, is right in this area. And contiguous that is the atrial, the membranous septum, which the tricuspid valve actually bisects into the atrial membranous septum and the ventricular membranous septum, as he abbreviates AMS and VMS here. So completely taking away the right heart here, we see the aorta, The non-coronary right coronary cusp, this little triangular region is the right fibrous trigone, and continuous with that is the atrial membranous septum and the ventricular membranous septum. So I'll kind of back off so you can see the actual anatomy, and it's this right fibrous trigone that's continuous with the atrial and ventricular membranous septum here. So a very oblique cut showing the LV and the aorta in the middle and the coronary sinus, and right above it is a couple of red beads indicating the AV node. And extending from that will be the AV bundle that goes up through the membranous septum and will bifurcate on the crest of the muscular septum into the left bundle and right bundle. And kind of showing this as a cartoon from Ho and Ernst really shows that this green tricuspid valve is bisecting this membranous septum, and the AV node will course into the central fibrous body as the AV bundle, go across the crest of the muscular septum, and then branch into the left and right bundles. And I think I'd be remiss if, given the opportunity, I didn't mention the triangle of Koch as the border of the tendon of Todaro, the tricuspid septal leaflet, and the roof of the coronary sinus. So a couple of pedantic slides here about what is the cardiac conduction system. I remind you that the heart is a syncytium of cells connected electrically through gap junctions made of connexins. So if you just say the conduction system is the part of the heart that conducts, well, all the myocytes conduct, so it's an incomplete definition. So really, the cardiac conduction system is composed of specialized cardiomyocytes with primary purpose of orchestrating electrical activity rather than mechanical pumping. So these cells in the conduction system are myocytes. They do have sarcoplasmic reticulum and all that. They just don't work very well. So looking at some patterns here with the connexins, we see that the contractile myocardium has connexins 40 and 43, and the sinus node and AV node don't express either of those, but they express this connexin 30.2, which is kind of a low conductance, high resistance connexin. And then the bundles and the Purkinje fibers are kind of a hybrid of these two because of how they develop. So talking a little bit about the embryology of things here, I'm going to use the story of the embryology of the heart as kind of a scaffold to tell the story, because I'm sure we've all heard the story before several times. And in a couple of slides, I'm going to skip the early stages and go straight to the bilaminar disc. And then the development of the heart as a whole starts from mesoderm that forms the first and second heart fields, that then forms the primary heart tube, that undergoes looping, and then septation and valve formation. And then finally, the outflow tract forms to the pulmonary artery and the aorta. And it's the endocardium and neural crest cells that form the cushions that become the septations and valves. So with the story of the embryology of the cardiac conduction system, it's really kind of three different stories because there's three different tissues here. The sinus node arises from the sinus venosus. The AV node arises from a ring of tissue in the primary heart tube called the AV canal. And the ventricular conduction system starts heading towards a ventricular phenotype, but then changes course to become conduction system tissue. And the advances in this area in the last 10, 15 years have been advanced by molecular biology. So you're getting into a large list of genes involved here. And part of my challenge in learning this stuff was trying to not lose the forest for the trees. There's a lot of things that can distract you. And what I've going to present here is that some of the important players here for regulatory are TBOX3, NKX2.5, and PITX2. And the important functions are these four listed here that are actually controlled by TBOX3. The really fast conducting connexins 40 and 43, HCN4 that gives you the funny current in the sinus node, and the SCN5 that's kind of hallmark of the sharp upstroke of the depolarization of the ventricular myocytes. And the heart really is asymmetric everywhere. You know, 4 chambers, outflow tract, these are all different structures, different sizes, different shapes, different functions. And it's not too surprising that the genes involved in coordinating that asymmetry is vast and deep. And so really trying to only kind of focus on what I'm talking about today was a challenge. So skipping to the bilaminar disc here, after gastrulation, we get the 3 germ layers, the ectoderm, mesoderm, and endoderm, the primitive node and primitive streak. And the kind of the cranial third of this streak forms the right and left heart fields that then coalesce into 2 crescent-shaped first and second heart fields. And even at this stage, you can appreciate that all three axes of asymmetry exist. There's a left, there's a right, there's cranial, there's caudal, and there's an ectoderm and endoderm, so I guess a top and a bottom there. As the primary tube develops, you get the sinus venosus on the caudal end, you get the artery, the aortic artery and the arches on the cranial end. The primitive heart tube itself is made of primitive myocytes that all have automaticity and a poor contraction apparatus. And as that embryo grows and lengthens, the primary heart tube develops more of a working myocardial phenotype. This is kind of a rotated 90 degrees to really show you that Y shape of the two horns of the sinus venosus, the endocardium, the cardiac jelly, and the myocardium. So the left to right asymmetry is a little bit more detailed. The sonic hedgehog gene really kind of drives left to right expression at the organism level. And through nodal and PIDX2, this controls left to right asymmetry in the heart itself. And backwards. And it's easy to kind of conceptualize that, variations of situs inversus, dextrocardia and all that can come from incorrect expression of these genes that really express the left and right asymmetry. So in the embryo, we've got PIDX2 that's really driving the left side expression, and that'll come up when we're talking about the sinus node. But first, the caudio-cranial asymmetry is established by T-box5. And T-box5 in high concentrations really promotes SHOX2 and T-box3. Again, this is one of the key players that I wanted you to pay attention to. It expresses T-box3 in sinus venosus, and this T-box3 is required for sinus node formation. And ultimately for T-box5, with rotation, it becomes an LV to RV gradient. And in three-chamber species like a frog, they apparently don't express T-box5 or that gradient. So on the left, we have PIDX2, and on the caudal end, we have T-box5 and T-box3 that really is expressed in the sinus venosus. So in the sinus venosus, I would call it as an extracardiac structure. It does not form from the primary heart tube. And I base that on this, that the sinus venosus is NKX 2.5 negative, which means it doesn't come from either of the heart fields. And it's T-box 18 positive, which means it comes from mesenchymal precursors. So based on this, I would argue that the sinus venosus and the sinus node are actually extracardiac structures and not part of the heart proper as it develops. And under the absence of NKX 2.5, the funny current HCN4 is expressed in the sinus venosus. So the actual sinus node itself comes around from the absence of NKX 2.5 and under the expression of T-box3. And in mice that are deficient in this 2.5, they have kind of disorganized pacemaker activity, and this is ultimately a lethal deficiency. And it's the T-box3 that really inhibits this kind of working myocardium phenotype of these fast connexins 40-43. It inhibits MPPA, which gives you the atrial natriuretic peptide, and the fast sodium current SCN5A. And so just a reminder, the action potentials here, the SCN5A really gives you that more rapid upstroke during depolarization. And the funny current gives you that slow depolarization of the membrane that really gives you the hallmark of the sinus node of automaticity. So why does the sinus node form only on the right side? Well, that has to do because the PIDX2 that's expressed on the left inhibits T-box3. So inhibiting the gene that causes kind of the phenotype for pacemaker activity drives the absence of a sinus node on the left side. And going a step further of suppressing PIDX2, in mice, this can develop dual sinus nodes and a form of right atrial isomerism in humans. And flipping the other way of injecting T-box3, you can turn atrial tissue into pacemaker cells. So this paper from the 60s showed a couple interesting things. This is of a chick embryo at the primary heart tube stage. And if you record transmembrane potentials at site A here in the sinus venosus, you get what looks like action potentials from pacemaker. And then at site V in the PHT, we see the sharp, very crisp action potentials that look like working myocardium. And then the surface at E here just kind of looks like the QRST that we're used to. And what I found interesting about this paper is that the pacemaker, the dominant pacemaker activity, as shown with the star here, kind of meanders a little bit throughout development and then ultimately ending up in the right sinus horn. And the real more interesting part is that the electrical activity precedes mechanical activity as indicated by the shading. So organized rhythmic pacemaker activity comes first and then mechanical contraction of the heart. So as the tube develops, you get more aortic arches, you get the cardinal veins, Bailin veins, and umbilical veins. The heart eventually undergoes rotation. In particular, the sinus venosus moves cranially and is more of a posterior structure. And I think this electron micrograph really demonstrates that nicely with the outflow tract, the common ventricle, and the common atria being a posterior and cranial structure. So one way that the embryo increases its cardiac output, because the heart is the first organ that actually develops and is functional in the embryo, is achieved through trabeculation. So as this lumen grows, the trabeculae grow into the lumen in hypertrophy, and perfusion of the trabeculae occurs through diffusion from the chamber blood. And it's not until the coronary system develops that you really get that compact myocardium on the outside of the heart. And in species that do not have a coronary circulation, they also do not have a compact layer to their heart. And interestingly, birds and mammals are the only groups that are dependent on the coronary circulation. So after rotation from the posterior here, we see a number of things are going to happen in the next month. A number of these caudal veins are going to obliterate. The right sinus horn invaginates into the right atrium, becoming the sinus venarium. The left sinus horn becomes the coronary sinus, and the left anterior cardinal vein will eventually obliterate and start creating the brachiocephalic vein across the top, ultimately giving you kind of the mature venous return to the heart at about a month later. So kind of breaking these parts down individually, the obliteration of the caudal veins leaves the IVC, growth of the brachiocephalic, and if the left anterior cardinal vein doesn't completely obliterate, it leaves what we call the persistent left superior vena cava. The anterior cardinal vein, if it obliterates properly, becomes the ligament of Marshall and the oblique vein of the left atrium, or the vein of Marshall. A thoracoscopic picture shows the ligament of Marshall crossing through the heart. And then on gross, you see the vein of Marshall kind of bounded by the left atrial appendage, the mitral annulus, and the left pulmonary veins. And this vein is becoming of more interest as it's recognized for its autonomic innervation inside of alcohol ablation in treating arrhythmias. The pulmonary veins grow as a common vein that then double bifurcates into the four veins. And at some point, this atrial myocardial structure has to transition to a vascular structure. And these micrographs show that pretty nicely of this myocardial sleeve extending into the pulmonary vein. And it's probably the anisotropy, the boundary, the anatomical boundary that plays a part in arrhythmia genesis. And also these pulmonary veins are very highly innervated by both adrenergic and cholinergic, and the autonomics are well established as provokers of arrhythmias. So isolating the pulmonary veins in early AFib is probably beneficial for these two reasons, and probably more too. But as the heart develops, the right horn invaginates, creates the sinus venarium. The sinus node will end up on the edge here next to the crista terminalis. The endocardium and cushions will form, septation and valves, and the septum premium will come down, the septum secundum will come down, ultimately giving this physiologic shunting in utero. And unfortunately, this is kind of where my story ends. I really can't find much references for why the AV node ends up right next to the tricuspid valve and how it ends up connecting to the left bundle and right bundle. And that as this interventricular septum grows from the apex to the base, I don't know this part of the story, so it's unfortunately just a question mark that I can't explain for you. But what I can share is that the AV node itself forms from this ring of tissue called the AV canal. And in this canal, it suppresses the myocardial phenotype through T-box3 and local notch inhibition. So a couple of experiments I want to show you that inhibit notch further or promote notch to see different changes in the AV node. And through further inhibition, we see that the AV node becomes hypoplastic, the PR interval shortens and this low conduction connexin 30.2 is expressed less. And through intracardiac electrograms, we see that the AH interval is shorter. And so flipping this around and overactivating notch, we see that you get rapid AV conduction with accessory pathway and pre-excitation. And that's shown in the, again, in the right diagram here, showing that the AV time has shortened significantly. And the surface EKG shows this slurring classic of a delta wave. And in this model, they were able to get conduction in mice, mind you, all the way to 1200 beats a second with 1-to-1 conduction. And they only stopped there because their pacing system wouldn't go any faster. So who knows how fast they could have paced it. And in 8 of 8 of these mice, they all showed accessory pathways, and 5 of the 8 showing at least a second, either left or right pathway on top of that. So the notch plays an important part in kind of balancing the function of the AV node, but also GATA4, as that promotes expression of that slow, low conducting connexin 30.2. And interestingly, there's kind of a teeter-totter with MyoR, that MyoR represses GATA4 and GATA4 inhibits MyoR. So it's kind of this balance that kind of fine tunes the delay. And I don't know if this is other genes or if it's physiological parameters or something that really fine tunes that PR interval. So moving on to the AV bundle. The AV bundle develops in situ in the AV canal. And neither the AV bundle nor the left and right bundle grow like angiogenesis or plant roots. They don't grow like that. They just form. And this ventricular conduction system expresses TBOX3, but somehow expresses connexins 40 and SCN5A that TBOX is supposed to inhibit. So there's part of the story that's not known of how it gets kind of this hybrid expression of these genes. Last is the Purkinje system. They arise from the ventricular tissue, but they have a really kind of poor contraction apparatus, and they have a really tight relationship with the trabeculae and not in the compacted wall. So you will not find Purkinje fibers in the compacted wall. So last section here, I kind of want to talk about the histology and function of the cardiac conduction system. And I want to start with this video just published a few years ago. That's from a high resolution CT model and demonstrates the components of the conduction system. In particular, pay attention to the AV node in green. And as this plays through, I'll kind of talk through it. So first, he's going to show you the right side and dissolve the RV free wall. And you can see the AV node, the AV bundle, and this little window here, that's the membranous septum, and really kind of how that AV bundle rides right up along that muscular bundle. And as he rotates it around here to the left side and takes away the left free wall, we'll again see that kind of window with the green riding that crest of muscular septum and giving off the left bundle. So here we see this window right here in that green AV bundle and the left bundle branching off over that crest. The last part here is going to be kind of a superior view, and it's going to dissolve away all of the myocardium, just kind of showing the conduction system and the aortic valve. And first off, we can really kind of see as this dissolves the separation between the left and the right bundle across that septum. But then the AV node and the AV bundle come out. The membranous septum is here, and then it kind of divides right at the crest of the muscular septum and divides into the left and the right bundle. So the sinus node is often described as kind of tadpole shaped, kind of in the saddle between the appendage and the superior vena cava. But a paper published 10 years ago now shows that the sinus node is not alone, and there's tissue adjacent to it that they called the perinodal area that, according to them, hadn't been described prior to 2011. And this tissue is kind of a hybrid of sinus and atrial myocytes, and it variably extends from the SVC down to the IVC. And this tissue is thought to kind of amplify the signal of the sinus node. And it may be responsible for some of the crystal arrhythmias being very kind of irritable tissue. And as shown again on that micro CT model, we see that, frankly, this is the ugliest tadpole I've ever seen, with all these projections and very irregular structure that I don't think tadpole is a good description for what the sinus node actually is. But we really see that the sinus node kind of hugs that boundary between the sinus venarium and the crista terminalis here. The sinus node itself is the most innervated part of the conduction system by the vagal nerve in T1 through T4, the sympathetic chain. And it's that HCN4 that provides that funny current that really gives that automaticity to the sinus node. A word about the intrinsic heart rate. Often you'll see this Jose 1970 paper that described the process of using atropine and propranolol to really give you this somewhat linear relationship between intrinsic heart rate and age. And this paper by Kaczmarek in 2019 showed something I think was kind of interesting, that in people with inappropriate sinus tachycardia, some had an elevated intrinsic heart rate. And those are the people that really responded to ivabradine, both in terms of their heart rate reduction and in symptoms. So maybe measuring the intrinsic heart rate is a way to predict who's clinically going to respond to ivabradine. So connecting the sinus node to the AV node is the internodal tracts. Well, maybe. This has been debated for a very long time with some heavy hitters weighing in. The problem is I can't find any modern papers describing either the support or refutation of the existence of these tracts. So I just have to leave it at that. But I will say that classically the inferior tract is described as projecting from the sinus node down the crista, which actually kind of sounds like this paranodal area that was just recently discovered. The middle goes across the SVC to the intra-atrial septum, and the anterior crosses over to the left atrium. The AV node, which I've really been describing the kind of the compact node shown here in blue, this arises from the AV canal and really expresses this slow conducting 30.2, which really gives the hallmark function of the AV node of delaying the signal to the ventricles. But adjacent to it, as shown here in yellow, is the lower nodal branch, which is actually ventricular in origin and also continuous with the AV bundle. And each of the compact node and the lower nodal branch have these little projections that project inferiorly, called the leftward and the rightward extension. And depending on who you talk to, this rightward extension may or may not be involved in AV nodal reentry tachycardia. So as this compact node and lower nodal branch extend, once that crosses the central fibrous body and becomes insulated, becomes the AV bundle proper. And shown here, a nice micrograph from Ho and Ernst that really shows that this fibrous membranous septum is split by the tricuspid valve, and the AV bundle rides inside the membranous septum, insulated, and really hugs this crest of the muscular septum here. And so giving off the left and right bundles, both of them stay subendocardial, and they both traverse the septal wall on their respective ventricles. The left branches into the anterior septal and posterior. Yes, there are 3 fascicles. And the right branches into the moderator band. This is a drawing from Tawara back when he discovered the AV node, and really shows that very nice reticular Purkinje system with the 3 fascicles, the anterior septal and the posterior. And just to point it out, because I can, the right coronary cusp, the non-coronary cusp, the right fibrous trigone here is continuous with the membranous septum with this AV bundle running right along the crest of that muscular septum. A gross specimen from Ho and Ernst shows the same thing again. The right cusp, non-cusp, the trigone, the membranous septum, and the left bundle coming across onto the septal wall and branching to the 3 fascicles. This paper from Cabrera kind of shows the same thing, but they nicely illuminated the membranous septum here, and they stripped away the endocardium, so you really can kind of see the anterior septal and the posterior fascicles of the left bundle. This is an okay picture from Ho and Ernst that really shows that right bundle embedded in subendocardially and insulated. And this is a kind of a longitudinal section by Cabrera that really shows the left bundle sandwiched subendocardially between and insulated by these layers of connective tissue. And so at this point, if you thought that, there's the AV node, there's the AV bundle, you get a nice Y bifurcation in the left and right bundles, and that's the end of the story. Well, clinically that's true, but anatomically that's not true. And 40 years ago, Anton Becker's group described this dead-end track. And so the AV bundle branches to the left and to the right and then continues on as this dead-end track. And it can variably terminate in the central fibrous body, in the aortic root, or the ventricular summit. And so it's kind of hard to ignore the coincidence that these are the areas where we get idiopathic PVCs and idiopathic VT. But clinically, this is not recognized as associated with those. The other possibility, other thing, is that of the retroaortic node, which makes three: the sinus node, the AV node, and the retroaortic node. And in some species, these AV rings that go around the mitral and tricuspid annuli are better recognized on histology, but none of these have any known clinical relevance in humans. So to show that with pictures, here's the picture from Anton Becker's paper showing the right heart here on the left side of the screen, showing the AV node, AV bundle, the right bundle, and then these dots just representing the dead end tract that just stops and goes to nowhere. And then from the Anderson paper, we have, particularly on the right picture here, we have the atrial myocardium, we have the non-coronary cusp, part of the mitral valve, and this off-colored tissue here is the retroaortic node. And I also included this picture from the same figure that really nicely shows the AV bundle branching into the right bundle across the top and the left bundle across the crest of that muscular septum. So again, the dead end tract and the retroaortic node are not of known clinical significance in humans. So just to kind of show that in this cross-sectional view, the AV node is approximately here. Those AV rings would go around the annuli of the valves, and that retroaortic node would be hidden somewhere back behind the non-coronary cusp of the aortic valve. Last is the Purkinje fibers, but I don't have too much more to say about these because I've said most of what I found interesting. They really run subendocardial on the trabeculae and exclusively with the trabeculae. And they really have kind of a mixed phenotype in what channels they express. This is a nice picture from Hohenert showing the RV from a cow, showing this really nice reticular pattern that these Purkinje fibers do. And then this photo is not as impressive of the left ventricle of a sheep, but really kind of shows that these Purkinje fibers are subendocardial in those trabeculae. So some highlights of my talk. The asymmetry is important in the development of the heart, the conduction system, and as I've shown today, in particular, the sinus node. And advances in the cardiac embryology in the last 10, 15 years has been really due to the better understanding of the molecular biology of the heart. And I would expect that's where the further advances in understanding how the heart develops will be found. And lastly, the conduction system consists of three tissues: the sinus node, AV node, and ventricular conduction system. And they have three very different functions and three very different stories in how they develop. If you're interested in any kind of further reading, Anderson and Becker are the kind of classic names in the morphology of the embryology of the heart. And other people such as Christoffels here really get into the molecular biology of the embryology of the heart. And Helena Dobrinsky, which I've shared a couple of her papers, really focuses on the sinus node. And one of her recent papers that I found rather interesting is that her group found that the sinus node actually has a circadian rhythm independent of its neural input. So it will cycle heart rates throughout the day independent of the autonomic innervation. Pretty cool. Some of the papers that made it into the final presentation here. And I'll leave you with this picture to think about how much artistic license this artist took in kind of drawing the conduction system and what is inaccurate about it. So I will leave it open for questions from anyone here or on Zoom. Any questions from Zoom land?
Colin?
Yes.
This is Dr. Bailin.
Hello.
That's very impressive. You know, I think it's goes to show you how complicated things are. In terms of, you know, development of the AV node, do you have any perception of how sometimes you get nodal fibers on the left septum versus the right septum? And Is that related to the extensions that Becker and Anderson described?
Yeah, that's part of the embryology story that I think is missing, that I don't really know how, as the cushions form the septum and the valves, exactly how the AV node joins into that mess. I don't know if it's because of, you know, misseptation that gives us all the other things like ASDs and VSDs that also causes, or as a byproduct of those fibers being on the right atrium. Again, that part of the story is just missing, so I don't know, and I don't really know that I have a good answer for you.
Hey, Colin, Barry.
Hello.
So nice overview. I think one thing I would add in terms of what's coming down the pike, I think the newest thing has been single cell RNA-seq, single cell transcriptomics, in the developing conduction system, that has now cataloging, sort of how the system develops. I don't think anybody's put it together yet in a way that really sort of adds a huge amount to what you've already done. But I think that will probably, at least in animal models, sort of be the next sort of advance that's going to happen.
Sounds like you would need a lot of data and a lot of samples to really kind of put together a nice cohesive story, but certainly in that realm is where I think things are going to keep advancing. Tyler.
Does MKX 2.5 have any role beyond sinus node manifestation? Like what happens to it in the adult sinus node?
So one thing I did share is that the absence of NKX 2.5 is a lethal mutation. I can't recall offhand how it functions in other tissues, but I do know it does. Let me go. No. I was going to go back. I don't want to flip all the way through. I don't want subtitles.
I was thinking like sinus node dysfunction or something. If you can overexpress NKX 2.5, are you going to recover some form of electrical automaticity in the sinus node?
Yeah, let me pull this up. So, all these genes play, like the hand 1 and hand 2 are known to be involved in asymmetry. And NKX, is over here in the ventricle. I did see some readings that there may be some mutations in NKX2 that give you arrhythmias in Holt or Am syndrome, both Brady and tachyarrhythmias. So it's surely has effects beyond just the pacemaker itself, but exactly to answer your question, I don't know. Other questions or comments from Zoom land? All right, well, I guess thank you everyone for coming.